Assalamualaikum and a very good morning to Dr. Masrinda and Dr. Zaida. My name is Nur Alia Batrisyia and today I am presenting on in silico immunogenic evaluation of conserved B-cell epitopes from an S3 protein of multiple serotypes of dengue virus in Southeast Asian strains. Moving straight into chapter 1, I want to address the background and the problem statement. So dengue virus is a global health crisis, particularly in Southeast Asia. It consists of 4 distinct serotypes, dengue virus 1 to dengue virus 4. Transmission is primarily driven by female Aedes mosquitoes. Historically, the vaccine development only focused on the structural surface protein, specifically the envelope protein. For the problem statement, the primary problem is antibody-dependent enhancements or ADE. Our current vaccines options such as Batrisyia and Dengue have shown varying efficacy across serotypes, leaving the vaccinated individuals vulnerable to breakthrough infections. This occurs when non-neutralizing antibodies from the primary infection, in this case the vaccination, act as a Trojan horse. So instead of blocking the virus, these antibodies actually helps the virus to entry into immune cells, which can lead to dengue hemorrhagic fever and dengue shock syndrome. Also, we have to consider the regional burden because Southeast Asia is hyper-endemic due to our tropical climate and urbanization. Next, the surface protein are highly variable and mutate to evade our immune system. Therefore, this hyper-variability makes them an unreliable target. My study asked what are the conserved region within the NS3 protein, the predicted B cells within it, and which of it demonstrate as a safe vaccine candidate. So to address these questions, the objectives are to determine the conserved region of NS3, to predict the B cells epitopes that trigger the best immune response, and to ensure those candidates are safe via antigenicity and allergenicity screening. The significance of this study is its focus on addressing the method. In silico is efficient, cost-effective, and rapid without the need to handle live pathogen. The core of my significance of study is actually immunological safety. So by focusing on the B-cell epitopes, we can find the epitope that elicit neutralizing antibodies, which are the first line of defense that physically block the viral entry. So this way, we can mitigate ADE risk by identifying only the most effective part of the virus, and we can exclude the region that trigger the harmful responses. So why do I target NS3 instead of the outer shell of virus? Because choosing NS3 protein is to bypass the hypervariability of the envelope protein, because NS3 is genetically conserved. So by the end of the study, the 3 outcomes expected are study determines the evolutionary conserved regions, the identification of specific B-cell epitopes within those regions, and hopefully select the high potential vaccine candidates. In Chapter 2, my literature review further established Asia as the primary reservoir for global transmission, accounting for 76.3% of imported cases, especially the 5 countries which are Malaysia, Thailand, Vietnam, Indonesia, and the Philippines. There are also clinical challenges where the overlap COVID-19 and dengue virus symptom leads to frequent misdiagnosis. Moving on to the advancement of the vaccine development, traditionally creating a vaccine takes 10 to 15 years and it carries significant biosafety risk which involve handling live pathogens. The example of the risk can be seen with the Qatar incidents. So by utilizing the reverse vaccinology approach, which is not only rapid and cost effective, but it also eliminates the risk of accidental infection during the research phase. Moving on to section 2.3, we compare the structure and the non-structural proteins. So traditionally, vaccines target the envelope protein because it is on the surface. However, the envelope protein mutates rapidly to hide from our antibodies, and it is actually the primary trigger for ADE. Moving on to 2.4 section, which is the NS3 protein structure and function. The NS3 protein is a 69 kDa multifunctional enzyme, and it contains a protease domain for cutting the viral polyprotein and a helicase domain for replicating viral RNA. So these functions are essential for the virus to survive and replicate. The virus cannot afford to let this region to mutate frequently because it literally loses the ability if it does. So this is why the internal proteins are considered evolutionarily conserved, which make NS3 a highly conserved target across all 4 serotypes of dengue virus. Next, we focus on B cell because they are the factory for antibodies and long-term memories. And then by finding the consensus residues within the B cells, which are the parts that stays the same across all Southeast Asian strains, we can create a broad spectrum vaccine candidates. Next, To ensure I will only select the most viable vaccine candidates, I have established benchmark. Firstly, antigenicity via Vaccine 2.0, I will only use threshold above 0.4, which is the validated benchmark in immunoinformatics that predicts whether a protein sequence is likely to trigger a protective immune response or not. So anything below that will be discarded. Secondly, the allergenicity, I will utilize Epitopes and allergen FP because we have to ensure that the candidates are non-allergenic to avoid life-threatening anaphylaxis reaction. When it comes to the limitations, findings are based on probability of algorithm and are not absolute, while highly accurate tools like Vaccine and IDB rely on training datasets. Next, the study lacks experimental confirmation through wet lab, and finally, the quality of the findings are limited by the sequences currently available in public databases from the past 10 years. Next is methodology. My methodology is divided into 4 phases: data retrieval, epitope prediction, safety and conservancy screening, and lastly, physicochemical characterization. This is my research flowchart. I will retrieve 200 NS3 protein sequences from NCBI GenBank from 5 hyper-endemic nations, which are Malaysia, Indonesia, Thailand, Vietnam, and the Philippines from 2015 to 2025. And then I will implement quality control rule, where any sequence with more than 5% ambiguous residues will be discarded. This is to ensure that the predictions are not based on unknown amino acids. And then I will align those sequences using muscle algorithm to observe the genetic relatedness, and then I will check for low entropy region through entropy analysis, where a low entropy means highly conserved and unchanged region. Next, I will perform B-cell epitope prediction. To ensure high accuracy, I will use a cross-validation strategy using 2 tools, i.e. DB-Baby-Pred for structure-based prediction and ABC-Pred for sequence-based prediction. So only consensus epitopes that are identified by both tools will move forward. Next, I will screen for safety and efficacy using Vaccine 2.0 and antigenicity using Epitopes and allergen FP. The physicochemical analysis using XPC proparam is to assess the molecular weight, isoelectric point, and hydrophaticity. Finally, I will use PyMol to map the epitopes onto a 3D model to confirm that the epitopes are surface accessible, meaning they are located on the outside of the protein where antibodies can actually bind to them. This is the Gantt chart that I will be following in the next semester. So in conclusion, this study will provide a rapid and cost-effective pathway to a regional vaccine that is hopefully safe from ADE and effective against all serotypes. These are the references used in this slide. Thank you for your time.